Our story begins in the late 1970s when scientists first dipped their toes into the field of genetic engineering. This began the race to find methods of efficiently altering genetic code. Among their first subjects in the early stage of genomic alterations were mice and yeast.
Scientists transformed yeast cells by using regions of genetic code that matched the genetic code surrounding the desired knockout gene. In this way, they were able to replace a gene with a selected tag.
Live mice were less important than their embryonic stem cells. With mice, scientists furthered their research by proving that an unfavored gene can be deleted by making changes to the cell in areas that favor said gene.
Our story continues in the 1990s when scientists discovered the double-stranded break, which introduced the age of editing. In this process, both strands of DNA are broken, and the cell automatically jumps into repair mode in one of two efficient ways. Scientists decided to exploit this natural process.
One method of cell repair is non-homologous end joining. The edits produced by NHEJ, small insertions and deletions, or indels, at the side of the cut, do not use a homologous template.
The second method, homology-directed repair, or HDR, uses a template to repair the DNA. In this situation, scientists can insert their own template to produce desired changes in the genome.
In the early 2000s, more cutting methods were created. These included ZFNs, TALENs, and the CRISPR-Cas9 enzyme. The first method is called zinc finger nucleases, and boy are they hard to make. These consisted of a zinc finger array and a cutting tool called Fulk1. Zinc fingers are small proteins that recognize and bind to 3 DNA bases, which provides a variety of cut sites. When Fulk1 is connected to the fingers, the DNA can be cut precisely.
Following ZFNs, transcription activator-like effector nucleases, or TALENs for short, debuted in the gene editing field. The tail array was made of naturally derived proteins that could bind to DNA, but unlike how zinc fingers covered 3 bases, each single tail only bound to one single base pair of DNA. Tail arrays, when connected to Fulk1, become TALENs. 2 TALENs together are then able to cut DNA.
Finally, CRISPR-Cas9 is born. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, which are series of recurring bases in genes. When paired with Cas9, CRISPR-associated protein 9, the CRISPR-Cas9 complex is able to cut DNA at a specific site, giving opportunity to change the genetic code at this location.
Even more recently, base editing has been implemented to change DNA without creating a double-stranded break. This functions like an eraser in the genome rather than scissors, so changes like C to U to T can occur in a single strand without alarming the cell.
With all the ways we have to create genomic change, who knows what the future holds? Gene editing has already allowed for advancements in agriculture, medicine, and maybe even babies. As we rapidly progress in this field, it is up to society and scientists to maintain moral and ethical practices so we can reap the benefits of gene editing while preserving humanity.